All right, so let's go ahead and get started here. So first of all, that content check part 2, the one that you might have turned in last Thursday, but maybe ran out of time. I will make a pile, or there is a pile started here that you can add your assignment to before you leave today. I have a couple other announcements before we get going. I've been asked to share about different programs. So if you're interested in genetics, there's information on the genetics club. Right? That includes if you're interested in, say, things like genetic counseling. Right? I left the information there. You can find it on the slides if you want to know more. There's the B&B mentorship program. Right? So if you're interested in the B&B major, you can get paired with an upperclassman who's gone through the process and help guide you through what they think worked for them and might work for you.
What's B&B?
Biochemistry and molecular biology. Oh. Yes, that is a great question. I should actually explain what B&B means. Another one that's not on this slide is the Tri Beta Honor Society. So Tri Beta is a biology honor society. You don't have to have a certain GPA to join Tri Beta. You just have to go to so many meetings and events. That's meeting in this room directly after our class today. And if you can't make it, you can still sign up to be a part of the club. You'll have to go online at Gus Davis and look up email information for that. As far as our class goes, I put a study guide on Moodle because our exam is a week and a half away now. So I would take a look at the study guide. I would say the study guide is not all encompassing, right? But there are some really good questions and concepts that maybe help you guide your study, all right? I also went back through all the slideshows we've had so far and the new slideshow today and put the portion of the study guide that is relevant to those slides directly in the slideshow. All right, so if you've downloaded it to your own device, you might have to go back into the Moodle folder to access it, but it'll tell you which questions are relevant to those slides. I think that is everything I had to say today. Let's see, we'll talk about the scientific literacy. We'll have a discussion today what that is due, and that organelles worksheet was due before class. If you have not done any of these assignments I have just mentioned, I have built in a little bit of wiggle room. I don't think any of these assignments are technically due till like 1 o'clock today. So there's a little bit of time. And if I don't grade it immediately, you have a little bit more time there, which I will honestly tell you, I'm going back to back all day until about 3, so you have some extra time there. Any questions before we begin? All right, I did have some questions submitted. So what exam or when an exam or test is coming up, will we specifically review the material on the exam? My answer to this is yes and no, right? So before the exam, which the class period before the exam is next Tuesday, there will be some time in class that is just unstructured review time. You can ask me any questions that you might have about the content. I will answer to my best of my ability without telling you what's on the exam, the structure, whatever questions you have. I will also provide you, I call it a mini exam. It's ungraded. It's just a worksheet for you to work through to kind of get a sense of the type of questions I might ask. Again, I'm trying to do everything I can to help alleviate any test anxiety that you might have. So yes, we can review material. No, we're not going to review every single bit of material that might be on the exam, because there's a lot of material, right? So bring your specific questions to that class period. More on the content-related side, I had a question, what is the difference between a protein subunit and a tertiary structure, right? And my answer is not that much, right? They're pretty much the same thing. So we're thinking proteins. We talked about this last week. We're going to jump into proteins a little bit more before, or at the beginning of this lecture here. but a protein, a subunit, and a tertiary structure. The tertiary structure is the last structural organization, last level of organization that a single polypeptide can undergo. Right? Now when we get multiple polypeptides that have tertiary structure, that have already been folded, and they come together to form a larger protein, right? That's when we get quaternary structure, and that's where this word of subunit comes into play. So for example, this image on the screen, we have this quaternary structure, right? There are 2 pink parts and 2 blue parts, right? Each one of these parts, whether pink or blue, we would call a subunit, because each one of these parts is a single polypeptide in its tertiary structure. Right? It's just bonded with other single polypeptides in their tertiary structure. So I would say each one of these is a subunit. Now 2 of these subunits are pink, right? So they're going to be the same type of protein, right? So maybe I would call that the alpha type subunit, right? And the other ones are blue, right? So 2 different subunits, but they're the same type. Maybe I would call that a beta subunit. Proteins have different ways in that they name their subunits. We're going to brush the surface on that. Are there any questions about tertiary structure and subunits? I would not expect you to know the word subunit per se. I would expect you to know that quaternary structure has multiple polypeptide chains in their tertiary structure. I think subunit is just kind of a good summary word for that. All right. I have gone through the content check, the first one, part 1 that you did on Moodle, and took a note of common questions that you might have had, especially in that reflective portion that I had you do. There were 3 main themes that I saw up here, right? One was bonds, one was polarity, and another one was like monomers and polymers. I have linked resources at the bottom of this page. They are videos that are different from videos I might have shared in those additional resources at the end of other lectures that might help answer those questions. But by large, the biggest question I had was about ionic and covalent bonds. How do I tell the difference? And I just wanted to clear up what I would expect you to know there, right? I am never going to ask you the question, which molecules are ionically bonded and which are covalently bonded, right? Now you might be able to tell what these are, specifically if you've had chemistry in the past, but this requires knowledge of looking at the periodic table, knowing which ones are metals, which ones are non-metals. I briefly mentioned this. I would not expect you to know this off the top of your head, right? What I could ask, say we're talking about covalent bonds, is I could give you a molecule. I would say that it is covalently bonded, and I would have you draw that molecule and tell me if it's polar and show me the polarity. Same thing we did for that content check in class last Thursday, right? Now, if I were to give you this question, I would give you all the necessary information, right? I would give you the information on the periodic table elements, and I would give you any electronegativities so that you could fully answer this question, right? If we're talking about ionic bonds, I might ask you, like, given a molecule, so CaCl2 contains ionic bonds, right? Right away, I'm going to tell you this is a molecule with ionic bonds, right? Bonds between chlorine and calcium. And I might ask you, why does calcium give up its 2 electrons, right? And why would chlorine accept 1, right? Each of those are chlorines accepting 1 each, right? So a conceptual question like the one I have on the board. I'm not going to go over this answer right now, but if you want to talk to me about it, come to my office hours. Happy to go over these with you. But I hope this kind of clarifies what you would be expected to know, ionic versus covalent. Right now, if you're in chemistry and they're going over these same concepts, they might expect you to know more than what I'm expecting you to know. All right. Last Thursday, I left you all at a cliffhanger on this question. All right, I said in proteins, what are the monomers, what are the polymers? And then I asked you if we were to change the primary structure of a protein, would that affect its tertiary structure? Now I've given you some time to work through this question. I'm going to give you about a minute here just to review what you had written down last Thursday, and then we'll come and talk about this question. Also a good time if you have anything to turn into a file. Use those now. Perfect. Yes, that works. So Mira, when I was little, like third grade, I broke my leg. And I had a janitor at my high school, or my elementary school, that made me a cane that looked very similar to this. Like really ornate and pretty. So that's what it reminds me of.
I got it at the fair.
Yeah, that's cool. That makes sense. Yeah. Yes, a combination of like a little bit of everything.
Wait, what did you say? Oh, so that's why. Because then once it gives away the calcium has two.
Yeah, it was this one technically.
Yeah, no, it was hard.
Oh.
Well, if she had a question like this, okay, it's and it says it gives up to us.
Yeah, calcium has two extra.
I was like, oh. This is bad. The other one is when they share. Ionic is dry. And then that's like a ionic is non-metal. Oh yeah, and I got you. I'm like, okay, so
All right, let's talk about this question. We're talking about proteins. What are the monomers in proteins? Amino acids, yes. What are the polymers? Polypeptides, yes. Hopefully that information is ringing a bell. All right, I know Thursday was a few days ago. All right, still thinking about proteins. If we alter the primary structure of a protein, would that affect its tertiary structure? I see heads nodding yes. Does anyone want to explain why?
It could affect the polarity and then the hydrogen bonds that
Yeah. So remember, amino acids have those different properties. Maybe if we switched one that was non-polar to an amino acid that was polar, that's going to change how that amino acid responds to all of the other amino acids around it. All right, I have an example of this one. So if you've taken lab yet this week, right, you should be familiar with sickle cell red blood cells. All right, if not, we'll get to it this week. In a healthy red blood cell, right, the primary structure of the polypeptide has this glutamine amino acid. That is what it normally has. In sickle cells, this one amino acid, and just one, is changed to valine. Right? That single change causes a change in how the protein is folded, and then causes the protein, in this case a red blood cell, to look like this sickle shape. Right? So that's just a substitution of, or a change in one amino acid, right? Having that dramatic effect. Again, because it's all about how those side chains of an amino acid interact with each other to form these very complex folding structures. Any questions? All right, I have a couple more slides on proteins here. because I really want to reiterate that the structure of a protein dictates its function. Right? Proteins have so many roles that they play within a cell. All right? And they are very, very diverse. Right? Diverse in size and shape and function, in the chemical properties that they have. Right? But it's all about their shape. If their shape were to change, their function were to change. And I like this image here. Right? Think about these are two proteins interacting with each other. Right? One is likely like an enzyme, so it helps speed up a reaction. And the other is likely a protein that it is doing some sort of chemical process to. But look at their shapes. Right? They're like two puzzle pieces that connect together. If I were to change the shape of one of these, say this orange one here, right? If I were to alter that shape so it no longer has these two grooves, maybe it has three grooves, maybe it only has one groove, right? This purple protein is no longer going to be able to interact with it anymore, right? Because the shape of the orange one would have changed, right? And there are ways that the shape of a protein can change, right? One of which could be those mutations like in the sickle cell red blood cells, right? We change out one amino acid at the primary level of structure. It changes its tertiary level of structure. But there are also ways that proteins can essentially lose its structure. Maybe we're not substituting out different amino acids. Maybe we're not changing the primary structure. Maybe it's that this protein finds itself in a different pH than what it's used to, right? Maybe it's really used to a neutral pH, and now we expose this protein to a acidic pH, right? That can cause it to denature or unravel per se, lose its structure, right? Salt can do the same thing, right? So maybe if you are an organism that lives in fresh water and now you put that organism into seawater, right? That salt can wreak havoc on its proteins and denature it. Same with temperature, right? Often enzymes, if we expose them to warmer temperatures, they will denature, they will unravel, right? So proteins can lose their shape in certain situations. We don't want proteins to lose their shape, which is why our bodies work really hard to maintain constant conditions in our body. Right? But it can happen. And when it happens, because structure equals function, that function is going to be changed. Or it would be non-existent. It might just be non-functional. Any questions about proteins? Alrighty, then we are going to move into our last of our 4 macromolecules. Alright, so we've done carbohydrates, lipids, proteins. Now we're going to talk about nucleic acids. Right? So nucleic acids are molecules such as DNA and RNA. Right? Now they're also made up of monomers. The monomers in this case are called nucleotides. The polymers would be nucleic acids, or the DNA and the RNA. All right, now I'm going to give you a very broad overview of nucleic acids, simply because we have a whole unit dedicated to DNA and genetics, and DNA replication, all that fun stuff later on in the semester where we will reiterate some of these concepts and dive a little bit deeper. But broad overview for now. All right, so DNA, deoxyribonucleic acid would be the full name here. It tends to be double-stranded, right? That classic double helix, right? If you see an image of DNA, you almost immediately know that it's DNA because of that twisting feature. DNA stays in the nucleus, right? So it's never going to leave the nucleus of a cell. And it stores all of our genetic information, right? So every single cell that we have has a full complement of our genetic information. RNA, on the other hand, it's similar to DNA, right? It carries genetic information, but it's only single-stranded. So you can see RNA here looks like just half of a DNA strand, which it basically is. RNA has the ability to leave the nucleus, right? So RNA can essentially carry information from the DNA in the nucleus to the organelles that need to use that information, right? Because they can go across that nucleus. All right, so monomers, nucleic acids, or nucleotides, excuse me. Monomers, nucleotides. Polymers, nucleic acids. Let's look at the nucleotides in a bit more depth. So nucleotides have the same basic structure, whether they are in RNA or whether they are in DNA, right? They're always going to have a phosphate group, right? So a phosphorus surrounded by these oxygens. They are always going to have a 5-carbon sugar, right? So if you look at our sugar here, and all the points in this shape represent where a carbon would be located, they're always going to have this 5-carbon sugar. And they're always going to have a nitrogenous base, right? Now the nitrogenous base is going to be the part of the nucleotide that will change and give it its unique properties, right? So just like in the amino acids, where all amino acids have the same basic structure, but it was that side group that gave it its different properties of being polar or nonpolar, the same concept applies here, right? We're always going to have a phosphate group and a sugar, and it's like not nitrogenous base that's going to vary a little bit between each different nucleotide. All right, so if given this image, you should be able to immediately say, that's a nucleotide, right? Or if told that your molecule contains a nitrogenous base, a phosphate group, and a 5-carbon sugar, you would immediately go, that is a nucleotide. All right, so the same basic shape for either DNA or RNA, but there are some differences depending on whether we're talking about DNA or RNA. All right. So, for example, in DNA, right, all nucleotides have a 5-carbon sugar, but DNA is going to have a deoxyribose sugar. Whereas RNA, that sugar is going to be slightly different. It's going to be just a ribose sugar. Right? The only difference between the two is just ribose has an extra oxygen on it. Right? But because we're adding in a different atom here, it changes its properties ever so slightly. So ribose in RNA, deoxyribose in DNA, right? So the sugars can be different. The nitrogenous bases can also be different, depending on if we're talking about DNA or RNA, right? So in DNA, we have 4 different nitrogenous bases. We can have cytosine, we can have thymine, we can have guanine, and we can have adenine. But in RNA, we don't have thymine. Instead, we have uracil. Right? So there's a substitution in the type of base here. All right, so RNA, DNA, all similar in that they have nucleotides, right? But their sugars are different. And then one of our nitrogenous bases is different. All right, you'll also notice that there are other terms on the screen such as pyrimidines and purines. We'll go more into detail about those in our unit where we actually explore this further. All right, so we have our nucleotides. These are our monomers. We have to create our polymer. So we have to create our DNA strand, right? If we look at our nucleotides and how they are attached to one another, right? They are attached in a very specific way that makes up what we would call a sugar phosphate backbone. Right? If we were to look at multiple of these nucleotides, all attached, you would see the backbone structure, or the parts that are all linked together, contains of a sugar and then a phosphate group. A sugar and a phosphate group, a sugar and a phosphate group, a sugar and a phosphate group. All right? That is our sugar-phosphate backbone. The nucleotides, right, they're not doing the linking between, the nitrogenous bases are not doing the linking between the nucleotides. Rather, they're just projected out to the side. Right? But that backbone, what's actually linked together are these sugars and the phosphates. Right? And again, we'll dive into this in more detail later on. But if we were to look at it just a little bit more detail now, just like a precursor to our Unit 3. If we look at a sugar, right, so this is deoxyribose in DNA, we can number the carbons within that sugar. Right? So starting on the right-hand side, we have 1, going down we have 2, and then 3, and then 4, and then 5. And if we were to look at how these sugars are used in creating this connection between the phosphates and the sugars here, right? It's this 3' carbon, so number 3 carbon, and the 5' carbon, right, that are going to attach the nucleotides to one another. Right, again, in Unit 3, we'll see why 5' and 3' are important when we're discussing like DNA replication and the direction of which DNA replication occurs. All right, so our nucleotides stacked together, we have our nucleic acids, either DNA or RNA. The actual process of linking them together is not unfamiliar, right? This is the same process we've seen. It's a dehydration reaction, right? So again, we have our 2 nucleotides, right? When they combine together, a covalent bond is formed, and a molecule of water is released. Right, so if I'm looking at this and I'm trying to determine, is this a dehydration reaction or a hydrolysis reaction, I would see this molecule of water as the product. Right? Essentially, I have made water. Right? So that would tell me that it's a dehydration reaction. I am taking water away from the original molecules. Notice that was also a common theme I kind of saw throughout those content checks as well. Right, so dehydration reaction, we are no strangers to these, right? It would specifically cause a phosphodiester linkage, which is again the type of bonding that occurs, covalent bonding, but specifically between nucleotides. All right, so we've bonded many of these nucleotides together to create a nucleic acid. Right? If we have one strand of DNA, right, DNA is composed of 2 strands, we have to have the second strand of DNA, right, and those 2 strands, right, those 2 nucleic acids have to join together to form the double helix, right? And here we see where hydrogen bonds, again, are extremely important. So if we're looking at our DNA strands, we have 2 of them here. So we can see 2 sugar phosphate backbones. Notice that the opposite DNA strand kind of looks like it's upside down. Keep that in mind. Remember that for Unit 3. We'll talk about why that is. In the middle, we have our nitrogenous bases. And if you can see, there's these little dashed lines between our nitrogenous bases. Those are all of our hydrogen bonds. Right? So it's the hydrogen bonds keeping those 2 strands together, keeping that double helix shape. Right, and they always follow this base pairing rule, which again we'll get into later. But adenine always binds with thymine, guanine always binds with cytosine. Has to deal with their shape, and that's why they're always paired together. Definitely feeling like 3rd week energy from all of you now. We're getting a little bit more tired as the semester progresses. All right, let's do a review to keep us awake and alert here. So for each of these molecules, I want you to tell me, right? Is there a monomer, right? And if so, what is the name of the monomer and what is the name of the polymer that it creates? I'll give you a few minutes.
What's that polymer again?
The monomers are monosaccharides.
Oh, is that one? It's like a more complex sugar. Proteins. Is there a similar term to that?
I don't think so.
That's just that's what I'm just
Monosaccharides.
How do you spell that?
It's not a fun spelling.
Wait, am I supposed to be able to spell that one?
I would say spelling for me, as long as I understand what I'm trying to say.
Wait,
You can probably get it.
Differentiate it out from other terms.
wait, mono. I feel like if you spell that,
I know.
So like that is. Yeah, that's the other two C's that have monos.
There is two C's.
So the monosaccharides and then the dyes.
That's why I don't expect you to.
Is it the dyes, fatty acids, and then the dye, and then
Yeah.
phospholipids also.
Well, the fats, there's fats about
Yeah. So monosaccharides are like so.
Oh, you're right.
What's the monomer for fatty acids?
Pretty small molecules compared to all the others.
Maybe the difference.
They don't have monomers. They don't have polymers.
Steroid.
I mean, they are made of fatty acids, that's why sometimes the plastics, sometimes radiocarbons, if we're talking about steroids. We just got carbohydrates. And they're not
And protein.
Are steroids polymers?
Can't get away with yeah. What are you going for? That's gonna be no problem. Either or.
No.
Stop.
Sorry.
Imagine. Sorry. What about nucleic acid? I just did that. Full fats.
It says amino acids and
Nucleotides.
Amino acids and polypeptides. And then polypeptides.
All right, let's review this together. Do carbohydrates have monomers? What are their names? Monosaccharides, yes, which makes polymers, which are called polysaccharides, yes. All right, what about lipids? Do they have monomers? They do not. Yeah, because lipids, again, they're relatively small molecules. They're not made up of the same repeating units, even though they might have multiples fatty acids. Might have 3, might have 2, right? They're not made up of these large chains of molecules. They do not have monomers. What about proteins? Yes, what is the name of the monomer? Amino acid, which makes polypeptides. And what about nucleic acids? They do, yes, and they are nucleotides, which make nucleic acids, right? DNA and RNA, yeah. All right, any questions about macromolecules? Because we are about to change topic. All right, well, I'm going to give you a breather from content. I actually wanted to give us a chance to talk about that scientific literacy assignment. All right, so I don't like it when I just give you an assignment and then we never do anything with it. So I want us to have a conversation. I have 10 minutes earmarked for this. Answer some questions up on the board here. They're likely questions that I already asked you in the assignment. Talk with each other, and if anyone doesn't have a group, look around you and add them to their group. This can be a bigger group discussion. And then after 10 minutes, we'll come back together and talk with each other. Also a good time to get up and take a walk down the hallway if you feel like you need an extra boost of energy. I know we're right before lunchtime. Well, I guess it's only 10:30, but
It feels like it should be lunchtime.
it feels like it, yeah.
Seems like
Because there are people in my grandmother, you read a headline saying, oh, we're diabetes, diabetes. And then it's not true.
that's what I thought. That's
Yes.
Bougie, that's what they say.
Fully fancy.
Yeah. Okay, you know what? This is relatively
Harvard research also gives us but not putting
Are you wearing Glossier? Are you wearing Burberry or Glossier?
Yeah, that means yes, kind of. Yeah. How long did it take you? I did. Oh, I haven't done it. No, it's not just the last class. There's a part of the language that we're going to
Russia. The article is like, so that's like.
And I,
Oh, that's one in your room? That's not this.
no, I'm saying it has this part of it.
Oh.
That's what that's.
That's
Wait, you have I was like, it was the brainwave is just not accident. People doing a lot. Okay, alright. That's spoke. You said it like it was a TV show. Like a movie. I've seen it. I don't know if I can quote it. I don't know. I'll see it. Yeah, I suck at watching movies. Like, if I don't watch movies with people, I just watch it.
Let's watch it with my friend's daughter. My roommate.
Okay.
We're just gonna join her.
Alright, wonderful.
It's actually chill downstairs.
Yeah, but that's what I'm saying. Like, at the end of the day, like,
This is what we're doing right now.
it's not like we're doing anything. Do you remember what we're doing?
I'm just kind of confused because it's just like fly fishing by now. I'm pretty sure I know.
Although fly fishing does kind of sound
Or like this side, like left side.
Yeah, like the teacher's garden just out there. But I'm pretty sure I ran to get a picture of Aaron is really good. Oh, yeah. I told my roommate she should come next time. Because I was giving her all the questions. Yeah, yeah.
The one that the side.
He doesn't know. Oh, it's on there. It's not like she's like
Yeah.
my roommate. They're literally all my friends.
That's a theory.
Because I would dump if I knew what it exists and it's
Yeah, so it's like.
Is there 7 of you in here? Okay, 8, that's like I was like, I feel like no, I know. And it's Hunter, Ryan, Kyle, Grant, Carter, Aiden. I couldn't remember. Okay, it was Aiden and Hudson, which are like the 2 guys I talked to. But I went on to so it's hard. I talked to them, they're like, why didn't you talk to me? Well, they just told me to like, what is that? And I said, I'm fine. And they're like, oh, okay, we'll do like message. So then I told them, and they're like, we're about to watch this. Do you just want to watch? I was like, oh, sure. So it's just so like random because like I literally talked to the least.
Yeah. But I know that's gonna help. Just making sure.
No, it's fine. Plus, like, since I learned before, like, seeing you guys' friends, like, I already met you. Because I met you. Wait, did I meet you before I was in or after?
It was after we got our cup.
So I didn't actually, but I did meet you before. We've been in the same room.
Yeah, on the same baseball field.
That time. No, that's That's so weird. I don't recognize you from the same I was sitting right next to you. I was sitting out around you.
Yeah, I'm guessing the little circle one. That was like right there where I was for you. That's great. I feel like probably I would have like remembered your face, but I don't know how I don't. I'll probably picture.
From the bank.
That would be hilarious.
I'm pretty sure.
Yeah, we took the picture after like this.
Finally after. That's really cool. The only person I remember from this year
Yes, for Jordan.
he got MVP for most funny kid. He won something crazy. Yes, he was MVP. That was the last person I was expected to win. Did you know he's it's not really what I did. Is that weird?
All right. Scientific literacy, is it important?
Yeah, that's what I thought too.
No comments. Is it important? Yes. Yes. Everybody should be saying yes, it is definitely important. Why? Why is scientific literacy important? Avoid misinformation. Yeah. So you can tell what is misinformation. Why else?
There's a lot of fake news like everywhere, so to be able to determine the two is really important.
Yeah, determine what is true and what is not, like factually based. Anything else?
Yes, if you go into stuff like science, you're going to do a lot of research papers, and that's a different type of academics.
Yeah. Learning, so especially for those of you in this room who are going on to be scientists, or even nursing majors, right? And you want to read the relevant research behind a product, a medication, a disease, a new species you're studying, right? You want to be able to understand scientific literature and understand how maybe headlines only pick and choose what they want to have you hear, right? So you can go into the primary literature. All right, so if you're scrolling through social media, because I think that's how most of us get our news these days is social media, there's a lot of clickbaity headlines out there. What are some red flags to look for in a headline title?
Too broad, making like assumptions, speaking in absolutes.
Yeah, maybe it's very broadly applied, like this can help everyone.
Very bold, saying something that's not catchy or
Yeah, like a bold claim or something that is you're like, what? No way, right? Anything else?
The source that it's coming from, like the website.
Yeah, so beyond the title, just seeing what source is it coming from, right? Is this like a BuzzFeed article, or is this, some scientifically produced article, right? I would add to this, a lot of these clickbait titles tend to be very dramatic, right? Very dramatic about what they're trying to get you to click and learn about. All right, so what are some of these examples of misinformation that we have run across? Who would like to share some specific examples?
Diet drinks cause diabetes.
Yeah, I've heard that one. Diet drinks cause diabetes. Anything else? Yeah.
This one lady who would be like really old because she drank, I forgot what drink it was, every day.
Yeah.
Drinking.
Yeah, some lady that lived to be old because she drank one thing every day. Right? Not likely there's other factors going on into play there. Yeah, I notice we see a lot of these in like the medical field, like especially different diets. You might also see it like sports or celebrities, right? So something very sensationalized to make you go, wait, did that happen? All right, so then I had you read an article with some misleading information. So is red wine the one thing you can drink every day for a longer life? See, a lot of no's. Why? Why is this wrong?
For one, you're not going to be drinking enough for it to actually make a difference without like getting drunk and having other side effects. And then for two, the studies they did, they were also eating a bunch of other stuff, and it was just a mix of good diet. It wasn't just one.
Yeah. It wasn't just red wine, right? It was a mix of others.
The flavonoid, like the thing that made people like healthier, actually causes more mortality. Like in the end, like they all cause mortality.
Yeah, they all cause mortality in the end, the flavonoids, yep. What else?
The like data from the actual study, like basically proving like that association and not like causation.
Yeah. And I actually I brought up this article and highlighted a sentence in like the second to last paragraph here. Right? They specifically stated in this research paper that it is important to note that the causality of observed relationships cannot be established due to the observational nature of the study. So basically they're saying that yes, this trend exists, but there's so many outside factors that might come into play that they just simply couldn't account for. Right? This was not a closed system study. They did not stick people in a room and watch them throughout their entire lifetime and documented every single thing that they did. Right?
The study was also only on women, so we have no data from her.
Yeah. So while the research is sound, right, the study only used women as their sample size, right? So this article claiming that it's the one thing for everybody to live longer, right? Well, this study only looked at women. They looked at young women that were relatively healthy to begin with. Yeah. Any other comments, questions about this assignment? All right, we do one of these per unit, so the next one will be about misleading statistics that you'll learn about next. All right, let's dive into more content, shall we? So we're going to talk about cell structure and organization. So we've talked about atoms, we've talked about molecules, now we're going to talk about cells, and eventually talking about how molecules can pass through cells. Let me put my clicker back into place here. All right, so if we're talking about cells, right, cells can differ depending on which category of life that we are looking at. Right? Are we looking at a prokaryote or a eukaryote? Right? Prokaryotes tend to be smaller, and they tend to have a really simple cell structure. Right? If we were to, say, dissect out a prokaryote, right, you're not going to find a lot of different stuff in it. Right? It's very simplistic. And the biggest thing is that there's no nucleus. And we can kind of see that in this image here. Right? If you look at that cell, it looks like a lot of empty space. There's some DNA in there. There might be some ribosomes, right? But a lot of it's just a lot missing. Right? Compared to a eukaryote, right? A eukaryote has a lot going on. Not only does it look like there's more in the cell, but there's more diversity of that stuff in the cell. Right? So eukaryotes tend to be much more complex. The biggest thing that differentiates a eukaryote from a prokaryote is that it has a nucleus. Right? That is the biggest defining feature. If it has a nucleus, it is a eukaryote. And that nucleus is what holds the DNA. Right? There's also a lot of different organelles within eukaryotic cells. So organelles tend to be membrane-bound components that have these very specialized functions. Alright, so prokaryotes versus eukaryotes. No nucleus, has a nucleus. Let's do a quick little dive into prokaryotes, but the majority of this lecture is going to be focused on eukaryotes. Alright, so if we look at prokaryotic cells, we could further classify them into two groups. We could say they're either bacteria or they are archaea. Right? Both tend to be really small, right? Really, really small cells. Bacteria are very abundant. Right? Bacteria are pretty much everywhere. Right? Bacteria grows on pretty much every surface. It grows in other organisms. We have bacteria in our bodies. Right? The vast majority of bacteria are not harmful. Right? We only hear about the harmful ones, right? Because they are the ones that disrupt our lives. But we have a lot of bacteria that live inside of us that if we didn't have them, then we would feel really sick. So a lot of gut bacteria is really beneficial for us. Archaea, on the other hand, are not very abundant. Right? They are less common. Again, very small, and they tend to be found in very extreme environments. So when I say an extreme environment, I'm talking about like the polar caps or the geysers of Yellowstone, right? Really hot, really cold, really lacking in moisture, right? Those very much the biggest extremes that you can think about on planet Earth, right? That's where you might find these archaea. That's pretty much all I'm going to say about archaea. Bacteria, I just want to reiterate. I don't know why my clickers decided not to work, but we're going to work with it here. All right. Bacteria, again, tend to be very simplistic. I don't expect you to know all the terms on this slide, but again, I just wanted to reiterate that bacteria, no nucleus. Right? Same with archaea, no nucleus. They can have some ribosomes, right? They can have an interior with some cytoplasm, some cytosol. They could have other structures like flagella here, which can help them swim, or these pili, which can help them stick to other surfaces. All right, but if we were to look at this bacteria cell, and then look at a eukaryotic cell here, they look very different. Eukaryotic cell has a lot more going on. The biggest of which is that it has that nucleus. And inside of the nucleus is the DNA. They also have a variety of organelles. Right? So I know you watched that video before coming to class that gave you an overview of organelles. But again, organelles are compartmentalized features that have specific functions. Most of them are bound by a membrane, right? Ribosomes being the exception. Right? And when I say they're bound by a membrane, they're bound by the same type of membrane that, say, a cell would have, a plasma membrane, or a phospholipid bilayer. Right? So again, we're using those phospholipids, we're bringing that back into play here. Now, organelles, they vary widely in what they do and how they look. Right? Their size, their function, their shape. They can vary depending on what species we're talking about. Right? So if we think, say, a plant species versus an animal species, plants will have chloroplasts that photosynthesize. We'll talk about those in a minute here. Animals do not. Within a single species, so if we think about like ourselves, right? We have different types of cells within our bodies. Right? We could have neurons, cardiac cells, epithelial cells, red blood cells. The organelles, which within each of these cells can be different. Right? Maybe they have more of one type, maybe they're lacking certain organelles. Right? It's going to vary depending on the cell we're talking about. All right, so organelles play a huge role in eukaryotic cells, but they're not the only thing in eukaryotic cells that we should care about. I forgot I had a group review question here. So I'm going to leave you on that cliffhanger. And we're going to pause, right? This is more just a review to make sure we're all up to speed before we're moving on. These questions might be relatively easy. I want you to answer these questions, and then we will continue.
They got them. And cytoplasm. And there's health. And they have membrane. Anything else? I feel like there's other stuff, but we haven't talked about this.
Both cells. They are. Prokaryotic are two membrane. It doesn't hold area. It doesn't have, say, like the nucleus, or it doesn't have like the endoplasm.
Nucleus being big.
What? When did you do that? At Subway? Those things are scary. You'll never get that last one.
But I'm gonna make it.
It wasn't like a Men in Black.
Yeah, you're right. Yeah, yeah. I like the reason
This is a reality one, right? Yeah.
No. Is that crazy to do?
All right, so let's come back together here. What is the main differences between the prokaryotic cells and eukaryotic cells? Nucleus, yes, that should be the biggest difference that you should be able to recognize, right? Which one doesn't have a nucleus? Prokaryotes, yes. So prokaryotes do not have a nucleus. Are there any big differences between the two that you thought of?
Nucleus. Wait, I said that wrong. How do you say?
Yes, nucleus, yeah, and? Oh yeah, they're lacking a lot of those organelles, right? They don't have things like the endoplasmic reticulum. No endomembrane system, which we will talk about in our next few slides here. So basically, they don't have a system of organelles. They have ribosomes. Pretty much it. So what are these similarities then? I kind of just mentioned one. They have ribosomes. What else do they both share? Cytoplasm. What else? What was that?
Membrane?
They have membrane. Yep, a cell membrane. What's another one? It should be, it's a big one. They all have it, but maybe in different locations. DNA, yes, they have DNA. Prokaryotes just don't have it in a nucleus. All right. So if we're looking at the cell, right, I know we've talked about, or I know that video has gone over some of these concepts, right, but I kind of want to reiterate some of these concepts. So let's talk about cytosol versus cytoplasm, right? Cytosol is the region inside of the eukaryotic cell, but outside of the organelles. So it's basically, you can think of it as all the fluid-like structures, the fluid inside of the structure, inside of the cell. Whereas cytoplasm includes everything, includes the cytosol, as well as all of the organelles.
cytosol, I say it wrong, that's the fluid?
Yes.
Oh.
It's basically like the fluid, yep. Whereas the cytoplasm is everything, right? So there's a slight distinction here. And cytosol isn't just the fluid, right? It does have some very important properties. For example, it coordinates a lot of what goes on inside of the cell. So it's a coordinating region for a lot of metabolic activities. Right? So cytosol is just the fluid. Cytoplasm is everything, fluid and organelles. The cytoplasm also contains something called the cytoskeleton. All right, so the cytoskeleton is essentially this network of protein filaments. All right, so the word filaments basically means long and thin. All right, so protein filaments are long and thin proteins. You can think of them like strings or ropes. There's different types of protein filaments. Right? I'm not going to ask you to know all of these, but I would want you to understand how they all function. Right? Microtubules tend to be the largest of which. Right? They're the topmost part of this picture here. They tend to be large. They tend to be hollow. Then we have the ones in the middle, which are appropriately called intermediate filaments because they are intermediate in size. Right? Again, they have a slightly different structure. They look more like a rope, right, than a hollow tube. And then we have the thinnest of the filaments, which are the actin filaments. All right, so we have all these different types of filaments. They're all working together to create kind of like this highway or transportation system within the cell. So you can think of as microtubules maybe are like the freeways. And the intermediate filaments are all of the roads that might go through a city. Whereas the microfilaments are maybe like the alleyways and the sidewalks. Right? They're all working together to coordinate movement within an area. Right? So they help with organization. So we can move substances or cargo along these highways, along these filaments. They help the cell maintain its shape. So it's kind of like a skeleton, right? The skeleton helps maintain our shape, right? It's maintaining the shape of a cell itself. And it can help organize things that need to move as well. All right, so we have the cytoskeleton. The cytoskeleton contains these filaments, but they also contain motor proteins. So motor proteins you can think of as like the cars or the bicycles or the people that are walking along these roadways and sidewalks. Right? The motor proteins, they function to basically move things within the cell. Right? Move cargo from place to place, help the cell bend. Right? They have a variety of functions. If we look at the anatomy of the motor protein, they all have this basic anatomy. Right? They have a head, they have a hinge, and they have a tail. Right? So at the top they have a head, they typically have two heads, they have a hinge region, and then they have a tail. So if you were to think of an analogy here, right? If I was a motor protein, right? I would be walking along, maybe say like a sidewalk, right? The sidewalk would be the cytoskeleton. My legs would each be one of these heads, right? That's the part that's going to move, right? It's moving along the hinge, which I could say is my hips, right? And then the rest of my body we would say is the tail. Now I know that's kind of backwards thinking, the rest of my body being the tail. This is not a tail, but it's like the rest of the motor protein, right? So it's going to kind of walk along the sidewalk, bending at that hinge, moving the motor proteins. All right, I have a video that I like that illustrates this. I think it's a very cute and fun video. They're going to look at a motor protein moving along the cytoskeleton in a neuron, right? But this concept can be applied to all other types of cells as well. I'm actually going to dim the lights here. There we go.
Our brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon. This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell, and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axon. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place. To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are two kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now. Fate strikes again. The other companion, Dynin, wakes up, and he can only walk in the opposite direction of John, resulting in a tug of war. But there can be only one. Along the axon in which John travels, there are places called synapses. Here, the axon connects to receivers of the other cells. Regulating proteins call the shots here. This traffic police makes sure that all passing traffic gets to the right destination. If John's cargo is needed in this synapse, he will be stopped and myosins take over his load. But today, John's cargo is safe. But what he does not know, that his road is under construction just a few blocks away. In our nerve cells, the cytoskeleton is changing constantly. Roads are built, but are also broken down. Facing this kind of obstruction, John has to find a detour. John isn't the only motor protein on the road. There are many more. Our nerve cells need a smooth traffic flow in order to perform well. A traffic jam due to problems during the journey may ultimately result in brain disease. Understanding the challenges John faces could improve treatment or prevention. Finally, John arrives at his destination. He has fulfilled his destiny. But several other Johns are just getting started.
All right, so I like that this is a good visual representation of the cytoskeleton. Again, there's many different types of motor proteins. We're talking about them very broadly in this class, but you might have noticed they said there are motor proteins like dynesin and myosin, right? We won't talk about those specifically. What I did want to mention, and let me turn the lights back on here, is that there's other types of movement than just the walking analogy. All right, so the video showed John, he was walking along a microtubule, right? There is another type of movement where maybe John doesn't move himself, right, but moves the filament under his feet. You can kind of think of it as like a treadmill, right? You're moving, you're walking, but you're staying in the same place. What's moving under your feet would be like the actin filaments or the microtubules or any type of filament. So it's kind of like the heads of the motor proteins are like grabbing the filaments and pulling it down, and then grabbing and pulling it down. So it's pulling it underneath of it, right? So in that case, the filament moves, the motor protein stays put. There's another example where both the filament and the motor protein don't move themselves, but rather they kind of make each other bend, right? And I was trying to think of an analogy in my class before this for this type of movement, and the best one I could come up with is like a game of tug of war, right? You're trying to pull the rope from your partner. Neither one of you want to stumble forward or move backwards, right? You're holding your ground, but you might be bending back and forth depending on who's stronger, right? That's the kind of movement I'm talking about for this third one, right? This bending type of movement. All right, so again, there's always variations and always exceptions in biology here. It's always more complicated than what it seems like up front. So that gave you a glimpse into kind of that complication. I want us to answer this question here. All right, it's almost Halloween, right? It's just around the corner from Halloween. I want you to explain why this image is accurate and why this image is inaccurate. And I'll give you a few minutes. One of the ways is it keeps the organelles from going. About how it's
Well, I'm saying because that they, like the way it's like.
Those are our organs are like. Okay, let me see how we can the fact that if you have a certain organ or something up here, like us, if your doctor sees their organ has to take out like your intestines no, I mean like when they finish the surgery, you just toss all your intestines back in. Yeah, because they move back. Yeah, on their own. But that's why you can't eat until like. So that's why you don't eat for a period of time. Yeah. To wait until all your intestines hang out. Yeah, they're alive. They do that when dudes want to straight together. Oh, but where?
Like there's a situation where it's like not just setting the same spot.
Do you know what language that is on the board behind us? Oh, I was like Spanish, right? Soy.
He was holding
Yeah.
like one of these screens for some reason.
Oh, I never took Spanish. I'm still telling you. I know, and I should because it's such a widely used language.
Are you allowed to take classes? Like for free?
It's like a great question. probably honor the class, but I don't think I would have time.
I know that like workers in the cafeteria can play basketball.
Yeah.
I'm assuming.
Yeah.
But I don't know where you find it.
Yes, that's the problem.
Night class. Yeah, because you want to go from teaching classes to going to no.
Yeah. I took Swedish when I was here. That was mine.
Okay.
Don't remember most of it.
Hey, you know what? Pass them.
All right, we have this image of this skeleton just chilling, right? Holding the entire cell together if it was the cytoskeleton. Why is this statement accurate?
Because it adds structure to the cell.
Yeah, it adds structure to the cell. Did we think of any other ways in which this statement is accurate? Honestly, I think we have the biggest one. It holds its structure. Which ways, how is this inaccurate? It's not static, right? Our skeletons are our skeletons. That's not going to change, maybe grow as we get older, right? From like toddler to adult. Cytoskeleton is constantly changing, right? Building new parts of the cytoskeleton, getting rid of the parts that don't no longer need to be used. Yeah. Are there any other statements that we could say about this in which it is inaccurate? I might add it's inaccurate again, not that it's static, but it acts as like a highway, right? It's the structure by which motor proteins can move. All right. Any questions about the cytoskeleton? Let's talk about some of these organelles then. All right. Now I had to watch a video for organelles. Right, and we're going to review the concepts in that video. I did want to mention that the organelles are all connected through an endomembrane system, all right, meaning that they all kind of work and function together. Sometimes that means they're physically connected. Think about the rough ER. Ribosomes are physically connected to the endoplasmic reticulum, right? Sometimes they're just coordinating with each other, right? So if we have, say, this rough ER here, maybe a piece of it buds off, becomes this vesicle, and transports materials to another organelle, such as the Golgi apparatus. Right? So they're all working together. Now I had you look at a variety of organelles in that video. And that video talked about all of these organelles except for lysosomes. So I want to quickly go over what is a lysosome, and then we'll review the rest of the organelles. All right, so lysosomes. They are organelles that break down complex materials, right? I think I heard trash can being said. Yes, they're kind of like the trash can. You throw things away in them, right? But then it takes it a step further and breaks down the compounds. So maybe we could say it's like a composter. Right? These lysosomes, they contain enzymes that are specific to breaking down complex materials, right? These enzymes perform hydrolysis reactions, right? So remember hydrolysis, we are adding a molecule of water into the reaction in order to get that bond to split, right? Opposite of dehydration reactions. Right, now there's many different types of enzymes that lysosomes can carry, right? But they all have the same basic function. They break things down. Now lysosomes are kind of unique in that they're formed by budding off of the Golgi apparatus. Right? So part of this Golgi apparatus, the plasma membrane, it'll essentially like pinch itself off, and what's left is like a sphere of plasma membrane. Right? It might have things that need to be broken down, like pieces of food. It'll merge with some enzymes, and then we do the digestion inside the lysosome itself. Alrighty, so we have lysosomes. I want us to review all the other organelles we learned about. I realized in my last class, I don't expect you to know peroxisomes, but I have in years past, so it's kept on this slide. I forgot to get rid of it. So by process of elimination, you can figure out what peroxisomes are. But I'll give you a few minutes here to work through these functions. Still imagine it up. Yeah. Going here. Good. We're looking at that list so we immediately know which organelles they are. Still working on it.
Wait, on the review sheet, are you gonna have like this?
Like the study guide?
Yeah, oh yeah, that, yeah.
Not necessarily. There's not gonna be like a list of terms you need to know, but like a list of concepts.
Yeah.
And you'd have to know the terms associated
Yeah, that's
with
like-
concepts, yes. Yeah.
Like I probably should know what a rough,
Yes.
I can't even say that.
And I'll say for these organelles, you should know all the organelles that I asked you to look at on the worksheet, plus the lysosomes that we talked about in class.
What about the bullet points for these questions?
No.
I really can't.
Yes. So like the nucleus, the ribosome, the endoplasmic reticulum, Golgi apparatus. These are all their basic functions.
Is above lipids where there's no slices that
No, it's not. Okay, that's what I thought you had, but that was here. So let's think about that one.
I'm also.
Going here.
Wonderful.
Yeah.
Coming back.
All coming back, you know all the answers?
No, not yet. I'm not there yet. And the vacuoles though.
Ah, there we go.
I got one. And lysosomes, we just did that.
Yup, yup.
What about the Golgi?
What are
either
Think of like a vacuum,
Any questions
Well,
here?
Which one is the nucleus?
Yeah.
So say what it says.
I did not put the
Think of it like when
opposite.
you're vacuuming.
Oh wait, no.
So what does the nucleus contain?
DNA.
DNA. And what does DNA contain?
just holds
RNA.
DNA or?
RNA, or information. Yeah. So it contains the information that might tell the rest of the cell what proteins are needed.
Okay.
Yes. I made that one tricky on purpose.
How they break down.
Wait guys, I think
That's this one.
it's got smoothest for lipids, like
Oops, I know your
All right, I get the sense that maybe more time might have been needed on this question. Well, let's review this together because we're almost out of class time. All right, I intentionally made some of them a little bit tricky just to kind of test you. But let's start with the first one. The location where lipids are synthesized. The smooth ER synthesizes lipids, yes. So that would be number 3 on my list here. What produces proteins? Ribosomes. Yeah, ribosomes produce the proteins. What coordinates this protein synthesis? It's the nucleus. This is a harder one. I did not put the obvious function for the nucleus on this list. Right? The obvious function is that it contains the DNA. It houses the DNA. Right? But that DNA contains information, and that information coordinates which proteins need to be when and made at a certain time. Right? So that would be the nucleus. What about hydrolyzes many biomolecules? Lysosomes, we just talked about those. What transports and sorts proteins? The Golgi apparatus. What stores materials? Vacuoles. Vacuoles have other functions as well, but we're going to focus on the storage aspect. That's its main function. Which membranes are used to aid with protein synthesis? It's not directly synthesizing proteins. The rough ER. Yeah, so there's ribosomes attached to the endoplasmic reticulum. All right. And so the endoplasmic reticulum provides a place that helps to make these proteins in conjunction with the ribosomes. And my last one here, aids in detoxification. This is one we didn't go over. I wouldn't expect you to know, but it would be the peroxisomes. The peroxisomes, no. All of the other organelles, yes. Right? So all the organelles I might quiz you on or test you on would be the ones that you went over on that worksheet. I have additional bonus slides at the end of this that go over all of those organelles as well. We won't actually go over those slides in class, but they might be a good study tool. Now I'm going to skip ahead and let you know that I have opened the next content check. That is actually due at the end of day Thursday. All right, normally it's due before class, but we're running a little behind and we're not going to get to some of those topics until class on Thursday. So I've extended the due date. You can start working on it now, but we have not covered all the topics yet. That's all I got.
Still not used to those.
Which ones?
Oh, the table.
Oh, the table.
You're like, what?
Yeah.
Can I clarify? No. You're doing great.
I thought you're like still not used to review questions. I'm like, we should be getting used to them by now.
The rough ER does still synthesize lipids, right? Or not as much as the smooth ER?
If it does, it's not its primary function.
Okay.
I have to research, does it synthesize lipids?
Something that it does, but like the smooth ER is more specialized for it.
Yeah, smooth ER is definitely more specialized, yep.
I was wondering if there was like a separate unit for like respiration and all that stuff.
There is.
That why we're
That
not
is our
covering it?
next unit.
Yeah.
See ya.
Did you get a chance to look at that
I
presentation?
did get a chance to look at it. I thought it was impressive.
Right.
Yeah, all the steps you did, the way you isolated them out. You'll do
didn't
very
get to
well in
actually look-
organismal biology.
Hmm?
You'll do very well in organismal biology. We do like some of those same steps.
Yeah,
Yeah.
well, I really want to do the fire program.
Oh yeah.
I think Eric was talking about that too.
Yes.
But like, yeah, I really want to do that. And then, but like, I didn't actually get to look at him under a microscope, which is the one thing that I really wanted to do, and that Dr. Franston told me would have been like the crux of it. I just ran out of time,
I mean,
so I could actually know what species it
makes
was.
sense. Yeah, it was very interesting the way that they actually grew better with gasoline.
Yeah.
Yeah.
Because they're probably metabolizing it.
Right.
But
right.
I couldn't say that.
Yeah. No, I thought it was nice.
All right. Well, thanks for looking at it.
Yes. Yes.